Hi, I'm Fiona McBride. I'm a senior lecturer in the Department of Physiology and I head the Cerebrovascular Research Lab looking at factors that regulate blood pressure and blood flow to the brain in health and disease. So right now my research focus has really shifted to looking at ischemic stroke. One of the major medical breakthroughs sort of in the last decade has been the advent of a procedure called endovascular thrombectomy. So it's a technical term, but all it means is the clinicians can now go in if you have a big stroke. They can put a catheter up your femoral artery, thread it up past your heart, up inside your brain and actually pull out the blood clot that's causing the stroke in the first place. And so I kind of got caught up in the excitement of that and actually thinking about some of the very real problems with, how do we manage the physiology in these patients, because we're going in to take this clot out, their blood pressure is going to be pushed down by the anaesthetic. And so it's not you can just let the body do what it wants to do anymore. You've actually got to choose somewhere to set that blood pressure. You need a target blood pressure because you're going to have to do something about it. You don't want it to bottom out. And so because there's this active decision, we really need to make the right decision. And maybe that's not the same in every patient. So what I'm trying to do with my preclinical studies is, try and unpick that black box a little bit and see what's actually happening to blood vessels in the brain, see if we increase the blood pressure a little bit, does that help? And we've got some really good tools to let us do that. My projects tend to be fairly ambitious, so I like to record lots of signals, and whether that's using one or combinations of different telemeters, two in one animal. So to give you some examples, in our stroke project we measured intracranial pressure, so we're kind of interested in what's happening in that intracranial environment. So we measured intracranial pressure and brain tissue oxygen, and those brain tissue oxygen electrodes, so we've got the telemeter here. You can see there's some really fine electrodes here, and these actually sit in the brain itself. So we make some little holes in the skull and kind of insert these in a stereotactic frame to really precisely place them into the area that we know is going to have the stroke later. And so that then lets us record the oxygen in real time. So these are really high resolution signals. We can record during beat-to-beat blood pressure, etc. We've got other experiments running where we're interested more in the flow aspect of the response, that relationship between blood pressure and blood flow. And then we can record those simultaneously beat to beat while we experimentally induce some decreases in blood pressure, for example, and we can record how the blood pressure drops and then what happens to the blood flow signal in that situation. So by combining kind of our specialist techniques with the instrumentation and the recordings, you can really start to answer some quite intricate questions about the physiology. They say time is brain, so when you've got an occlusion, you're losing millions of neurons and neuronal connections every minute that occlusion, that those areas don't have enough blood flow. So it may, it's a short window, but actually quite a profound potential for benefit to the brain if we can get this right.